So, thank you everyone for joining. My name is Julia, and I'm a scientific engagement manager here at protocols.io. And so, we are here for the tips and tricks webinar today, which is a more advanced webinar where I dive into some of the additional deeper functionality that protocols.io has. So, if you are joining today and you're very new to protocols.io, that's okay too, but some of the things might be, you might need a bit more context with one of our intro webinars first. So we do have new users joining sometimes. So in case that is you, I will put a link into the chat to one of the recent intro webinars that we did. Before I kick off, a little bit of housekeeping. So I've already pressed to record the webinar, so you'll receive a link to the recording as well as some slides after the session. Also, closed captioning should be enabled if you would like to use them. If English isn't your first language, you can also turn on translations via the captioning, and I'll show you how to do that on the next slide. Q&A will be at the end, so we'll have 10 to 15 minutes where you can answer your questions. Feel free to put them in the chat as they're occurring to you or into the Q&A as well. We've got a small group today, so I think it might be easier to do it in the chat. Also, if there is anything that comes to mind during the session, there will be the opportunity to provide feedback at the end. So if there is anything, hold on to that feedback and you'll be able to submit it. So if you do want to enable translations, you'll need to go to the bottom of your screen in Zoom and click Show Captions. You'll then be prompted to select the language that you would like, and then the captions will be automatically translated. So you'll be able to have that there to help you out. So I will get started by showing a couple of slides of background, just as a reminder for anyone. And then we will actually jump out of the slides and directly into protocols.io and do a demo of some of that functionality. But to start us off with some background, the idea is that there are two sides to protocols.io, which you might be familiar with. The private side of the platform, which is where we are an experimental workflow tool where you can easily document and track how your methods are evolving over time. And you can do that in a collaborative space with other members of your research group or also within global collaborations as well. And then the idea is that you start using protocols.io as you're setting up your experiment, working on that private side of the platform. And then it makes it a lot easier when down the line you want to share those methods publicly. And that's where you can publish them on the open access repository on protocols.io. And if we think about that publishing side of the platform and how that might fit into publishing things like journal articles and other research outputs, this is how it fits in. So here you'll see a little bit on the concept of modular publishing, and that's where all of the different modules associated with a piece of research, so that might be something like code, molecular structures, methods, they are all put into fit for purpose repositories and linked to the paper that might talk about the narrative of that piece of research. And we can see a really nice example of that here on the right, where they have linked all of their outputs within the data availability section of that paper. And we can see what those are here. So they've put their methods onto protocols.io. Aside from the publishing side of protocols.io, there is a lot that you can do to really increase the ability for people to interact with and reuse your methods. So if you think about something that's relatively static, like a Word or PDF document, the protocols on protocols.io are very dynamic and interactive. And then you can take extra steps while editing that protocol to increase that ability for people to interact with it. And I'm going to show you how to do that today. And so the benefit of that is that it makes it as easy as possible for you as the protocol user to interact with and use that protocol as you're working with it. And then it also makes sure that that protocol is a lot more clear so that it can be reproducible for other people and make it a lot easier for them to build on it. So I'd encourage you to explore what you can add into your protocol. Before I jump onto protocols.io, I will talk a little bit about some of the terminology that we use. And this is something that we do get some questions about. So when it comes to building on others' work or managing your own protocols, there are various ways that you can do that. So if you have a method that you've documented and you want to update that, you can do it via these different mechanisms here. So if you're creating a new update to a protocol, you can do so via a version, a fork, or a copy. I've got a little food analogy to help us talk about that today. So if we think about a version, if you are baking a cake and you might have a recipe for chocolate cake. If you improve on that recipe to make the cake more dense, more flavorful, more chocolatey, you might create a new version of that recipe because the recipe, it's still a chocolate cake recipe, but it's a new and improved one. And that's what you can think about as a new version on protocols.io. So on the platform, we see that the new versions are linked to the previous versions, so you can navigate between them here. And anyone can create a new version, so their own private version of a protocol. But if you create a version of someone else's protocol, you'll need permission from them to publish it. So only the original person who's published that protocol can publish a new version. And some of the reasons that you might want to do this in protocols.io is if you're rectifying an error in a previous step, or providing additional information, or as I was talking about with the recipe, making that protocol new and improved and better. What you can also do is create a fork. And that is where, if we go back to our chocolate cake recipe, maybe I'm going to adapt that recipe for making a vanilla cake instead, or a lemon cake. We've got yellow here. So I've taken that recipe and I've edited some parts of that recipe to make it applicable for a different recipe or a different scenario. And on protocols.io, that fork, it is linked to the original protocol that it was forked from. And so we can see this is if you publish one, this is the name of the protocol, and we can see directly underneath it what it was forked from. So this is to ensure that the developers of the original protocol can also be credited for other people building upon their work. And the reason that you might create a fork on protocols.io is if you are modifying that method for something like a different sample type, species, strain, a different piece of equipment or lab setup, if you have to adapt it for a more low-cost situation, things like that is why we see people creating forks on protocols.io. And then the last one that I'll talk about is a copy. And so this is a little bit different, probably not used as often. It's more about if you had a template of your recipe that you've written up that says it's been developed by, if I had a recipe that had been developed by Julia in my kitchen, and then I had all of the metadata associated with my recipe that I knew would be the same for a completely different recipe, but the recipe is completely unrelated. So it's essentially using the metadata around that protocol as a template. And so there's no link, there's anyone can So only the owner can create that copy because they're probably the only ones who will have the same metadata. And so we can see an example of that here on this slide where they had a collection of multiple similar protocols. They had different content, but for example, the authors were going to be the same for all of them. So, with that out of the way, what I will do now is move out of the slides and directly onto protocols.io. You'll see here some of the information that I'm going to be covering today. Because this is a more advanced webinar, there is some assumed knowledge, which you can see there, but we'll dive more into these topics. So with that said, I will stop sharing and then share on protocols.io. One moment. All right, you should all see my screen now. So I'm in my workspace on protocols.io. And if anyone is new and you want some guidance with getting started, creating a protocol, playing around on the platform, you can always open up. There's a little raccoon helper that will be at the bottom of your screen, and they'll instruct you on how the different ways that you can get started creating a protocol on protocols.io. But we're going to start off by talking a little bit about the sharing functionality and the different options that are available there. So if I go down to one of the protocols that I have in my private workspace, I know that if there are other people in my workspace, they'll probably be able to see that as well, depending on how it's been configured. But there is a lot of flexibility with how I can share that file as well. So if I click on it and I go in here to click on share, I can see who has the ability to see that protocol. So I, as the owner, can of course access it. And everyone else who is a member of my workspace, they have the ability to edit and publish. And I can come in and change that here for this individual file as well. So I am part of an organization on protocols.io. So this might be you if your university has a site license for protocols.io, and your organization would be whatever university you're located at, for example. And so I can have the ability to have this protocol be discoverable by anyone at my organization, regardless of what workspace they're in, and I can do that here as well. And you can see that I have already shared this file with a couple of people. So that's my boss, Emma, and I've made her a co-admin of this protocol. So she has lots of rights as well with it. Excuse me. And you can also, instead of sharing with individual users, you can share with a whole workspace. So if your workspace is in, if my lab, for example, is in this demo workspace, and I know that we're going to be collaborating very closely with another lab and they have their own workspace set up, maybe I would want to share this individual protocol so that it is available to the workspaces in both labs. And that file will appear in both workspaces. They'll be able to organize it within their own folders, however they want, but everything is synced across those workspaces. So if I do want to add anything more, I can click on Add More, and I can choose to share it with individual people or whole workspaces. So I won't go ahead and share it with anyone else now. I'll go ahead and exit out of that. But what we'll look at now is a little bit more advanced editing of a protocol and what are some of those really interactive components that you can add in. So I will move into my protocol. So I'm editing a protocol right now. And if you aren't familiar, make sure that when you're exploring, you check out all of the different things that you can add in. So we can add in overall structure to the protocol here on the left. There's different tabs with information along the top. And then, especially here on the right, I can add in things like components. And we have quite a variety of components. They are for lab-based research, field-based research, computational work as well. So there are a lot of different types of protocols that go on the platform. And I actually have usually an example that I maybe will find later and put in the chat that has a lot of nice examples. But for today, I will show you, and we've got this is a nice example of a computational protocol. And so we can see they've put in what's some preparation that you might need to do, what files do you need to prepare, and they've put in some commands as components, and we can see all of that there. And if anyone wants to check that out themselves, I will put this link here. And then another nice example of a more wet lab-based protocol is this one here. And we can see they're working with things like temperatures and amounts and times, and they have put them in as components so that they work really well with the run functionality on Protocol Studio. So now you can see what's possible. So we'll go back and add some of that into our protocol here. So if I'm starting out, I can come in and I can insert a step, or I can click here to add a step. I can also insert a new section. And then I come down and I can rearrange my steps by dragging and dropping. And then I can come down and I can think about adding some components. So if I have, I've actually already added this, what I'll do is, so if I had, for example, 5 mil that I was working with, I can highlight this and click on amount, and then it will convert that into a component that when I'm running this protocol can be, for example, scaled up and down, and have that calculation happen automatically. Other things like reagents, we have a reagent component, so I can add that in here and try and search for, we have a large catalog of reagents that have been pulled by various vendors, so I can link in the exact reagent and catalog number that I'm using in that step. Because we do know that there can be variability depending on what reagents that you're using, so it might be important to document that between different protocol versions or forks. And then here we've added in a timer, and I've added in a temperature as well. And we'll see a little bit more how these work when I move on to the run functionality. I've also added in some nice examples of some computational components that you can link to as well. And so all of those are found here on the right. So I think most of that should be relatively familiar already if you are an advanced Protocol.io user. But we'll dive in a little bit more deeper now. So if I'm wanting to attach a file, will now have to go to the top menu bar here to do that. So I can click and attach a file, and then I can upload from my computer, or I can also drag and drop a file as well. And you can see that I can attach that into the individual step where that file is relevant. I can also add in an image that will be at the top as well. And let's see, I normally have an image prepared. We'll do this one. And so I can come in and I can resize that image. I can add in a figure legend, center it however I want via these options along the top here. And then if you have a step that's particularly complicated and text in an image isn't enough to really demonstrate how to carry that out, you can also link in a video as well. So if it is a relatively short video, you can add it directly into the protocol step. And if that is the case, you would do that via this insert image, and then you would upload that short video. However, if you do have a longer video, and I've got the file guidelines here as well. So you can find this in our Help Center. So if there is any video that's larger than this size, you'll need to deposit it somewhere else like YouTube and then embed it into the protocol. And I'll show you the process for doing that. So I have this example protocol, and you'll need to go onto YouTube and click onto Share, Embed, and then you'll need to copy this text here. So not the URL at the top. So I will click Copy. I will go back to my protocol, and then I will need to go to Embed Media here, paste that code, and then we'll be able to see the video of that protocol, which I can play in that protocol as well. You can also add in tables to your protocol if you want to add some structure to the information that you're showing. So I'll put in a short table. So again, I'll need to go up to the top to access that. We are doing some updates to the tables, so they will be very much new and improved shortly. But for now, what I would recommend is putting in the title headings of your table directly into each of the cells. So I might have a amount, and then number of samples, and then actually I'll do this as volume. That's probably a bit more professional. So I can put that in, and just like in Excel, I can add in formulas as well. So if I know I had, for example, 10 mil, and then 5 samples, I can add in my formula. And so what you will need to do is type the cell references directly in. So I will need to type in A2 times B2. And it will calculate that automatically for me. And I can, if I have lots of things to prepare, I can add multiple rows, and I can also delete them and, for example, leave them blank. And then whenever someone's coming in and using that protocol later, they'll be able to fill that in and have that calculate automatically for them directly in the protocol instead of opening up a separate Excel file to do that. With a plate map as well, you can visualize that directly in Protocol Studio for how you might want to lay that information out. So you can attach it as a separate file like's been done here, but I will show you how to create one on Protocol Studio, and that is accessed again on the right here in Components. We can go to a well plate map. So I can add one in directly. These are some relatively standard sizes, or I can do a custom size. So I'll show you what that looks like to create it. But what I'm actually going to do, and what you can do as well, is import one. So we can see you have the option to import an Excel file. If you already have that information put, so it's going to detect the plate size. And then the information that's stored there is pulled across as well. And I can zoom out, or I can open this in big screen for that 96 well plate. So I haven't shown the file, but there is some color that was in that Excel file as well. So the color, unfortunately, isn't pulled across, but you can add in color yourself. So there is the ability to individually edit cells in terms of color and also typing in, replacing the text that's there. But if you have a 96 well plate, you probably don't want to be changing the color cell by cell. So what you can do is go into template settings and see what coloring options are available to you. So there are some templates there, but you'll probably want to create your own. So I have created this template, and then I can multi-select and create the template that I want. So I'm going to do that for this one and save. And then we can see that it's applied that coloring to my nice 96 well plate. So it's easy to understand what's what. Next is what we call a smart component. And this is where you can record a key value that should be documented during the experiment. So if you are someone who is relatively forgetful and you want to be able to be sure that you're noting that information down, this might be something like metadata for a piece of equipment that you're using that you want to record, or other experimental observations. That's what you can use the smart component for. So I can find that here on the right, and then I can add in what I would want to record. So for example, here I'm going to do again number of samples. And so I can leave this as optional, or I can mark it as required. And if I do that, which I am going to do, is we'll see when I'm running this protocol, it won't allow me to continue until I have filled in this information. So I can add in other rows as well, but I'm not going to do that today. What you can also do is, if you do have lots of different rows in here, you can download that as well. If for some reason you needed to use something other than protocols.io for recording that information. What you can also do is nest a protocol inside another protocol. So you can do that using the protocol component, which I have done here, and we'll find that here under protocol. Once you can search for published protocols or the protocols that are stored within your workspace. And it's a nice way so that people can preview those instructions when they're using it. And then in the run, it will also expand out, and I'll show you what that looks like later. But this can be very useful if you have, for example, a protocol that uses specific buffers, and then you want to link out to the recipe for that buffer so that if anyone doesn't have it on hand, they can know how to create it. This is what you might use that for. Or there's various other situations where you might have multiple protocols that you want to combine into one in this manner. And then finally, what I'll show you how to do is to branch your protocol. So most of what we've shown here is a linear set of steps, but oftentimes you might have a protocol where there's a set of steps that might depend on a specific variable that you would figure out during the experiment, or they would depend on what type of tissue or sample that you're working with. And in that case, you can branch the protocol and have a different set of steps for each of those situations. And so I can go in and I can add a step case. So that is here on the left. And then I might say, so for this example, I'm going to say that I've got different instructions for different cell types. So I might do liver cells. And you can add in further context to describe those different situations as you choose to. So I've got one case here. And then what I can do is, I'm on my small screen today, then I can add different situations. So I've got liver cells, maybe I want to do kidney cells as well. And so in each of these that I can select, I have the ability to add in different steps. And so then I can see that these are the steps that are associated for liver cells. And then I can come in here and I can see there's no steps added for kidney cells just yet. And then I will need to pick whatever steps that I would like to include. One thing to keep in mind is that when you do add in a step case and it does create that branch, once separated, it can't be brought back together at the end. So keep that in mind when you are adding in a step case, that you'll need to keep those different situations separate for the rest of the protocol. All right, so now we've added in some different functionality into our protocol, so we can preview it. I'll go click on View. And if we go down, we can see that most of it looks quite similar, but I'll show you as well what that step case looks like. So we can see I can then choose what my situation is, and I can see how many steps might be involved there. But now I may want to run my protocol. So I'm going to click on this Run button. And so then I can document how I'm following that protocol and what changes I'm making to that protocol when I'm carrying this out in the lab on my computer, wherever I'm doing that piece of work. And so we can see here, if I had put in some time into my protocol, it calculates that automatically at the top. And so that's coming from this 1 hour timer that I put in. And then I can come in, and as I complete step 1, I can mark it as completed, and it's going to document who's completed that step and when exactly it's been completed. So it's creating a record of how that experiment has been carried out. So I will move through these relatively quickly. But if we go down, actually, I'll go down a little bit further because that is what I want to show. But we can see I've come here to my smart component and I can't go to the next step. And that's because this information is required. So I'll need to put in some piece of information and then I can move on to the next step. But what I want to show as well is when I've nested this protocol, what's happened is we can see now under step 15, there's some sub-steps and that weren't in the original protocol that we're creating. And these sub-steps have come directly from this protocol that I have nested in. And so then I can follow those if I need to prepare my solution. Or if I already had my solution prepared, I could probably come in and click to skip these steps. But what I'll do is with this step, I will show you how to use the amounts and scale, for example, this step up and down. So we can see here, I can come in and I can make any edits that I might have made on the day. And if I go into settings here in this menu on the right, I can scale that step. So I'm going to scale it by 3. We can see because I've put that information into the amounts that it's going to do that calculation automatically. I can also, if it is appropriate and makes sense with the way I've laid out the protocol, I can also scale the entire protocol and all of the steps as well. And then because I've added in that timer component, that timer will run like a real timer as well. There'll be a little sound at the end, and that can be helpful too. So this run, it's stored in my workspace and it is collaborative, so it can be completed by other people if there are multiple people working on this method. And it remains editable until I click this finish run button. And then it's becoming an uneditable record of what happened. I'll go ahead and click finish. And then we can see I can't make any more changes here. Yeah. So let's see. Yes, so I can't make any more changes. I could add a comment later, but that's not changing the protocol itself, and it will be timestamped as well. So after I've created a run, after I've created my protocol, and after I've created my run, there's various ways that you can manage those pieces of work. So one of them is via the task manager. So if I go into my workspace, so for example, I'm in this workspace, I'll see in the menu this tasks. And when I click on that, it will take me here. You can see I've already created a lot of example tasks from past webinars. But if you're a postdoc lab manager, you might want to create tasks and link one or several protocols to those tasks. So I'll go in and create a task. I'll give it just this example name. So you will need to give it a title first and then click save. And after you do that, then all of the changes that you make will be automatically saved. So I can add in some context and a description and leave it at that if there are no protocols associated with it. But you might want to add in and link to a specific protocol. So the one I'm going to link to is this one, run test protocol. And what I can do is start to search. It will take a moment to search the protocols. Let me try that again. It's not happy with me today. If you do ever encounter anything like this, what you can do as well is click on this help. And then if you do have an issue, you can take a screenshot or a video recording and upload it. And the team's very quick at responding and rectifying if anything is wrong. Well, I will stick with this one then for the sake of the example today. Ah, there we go. Well, you can add multiple protocols as well. And once you've added the protocols, you can add in and assign them to people. So you can assign the whole protocol or you can assign part of the protocol as well. So I will say that maybe I will do the first half of the protocol and I can select when I want that to happen as well. Let's start with today. And I'm going to assign that to myself. And then I can add another assignee. This is my alter ego, my other test account. And then I can assign them steps 11 to 20. We'll let that load for a second. And so once I've done that, whoever comes and opens this protocol, they'll be able to see that. They can view the protocol or they can directly run it when they start that piece of work. And so let's say it will start today, and then it will last for 2 days, for example, and go on 22nd. So I've got a few dates saved here. We can see it's loading, and those changes have saved. And so we can see the people who I've assigned to this protocol, they've automatically been added to the list of assignees. But if I had, for example, maybe a postdoc who is going to be supervising these people but not carrying out the individual steps, I could add them as well separately here as an assignee so they would have visibility on what's happening. Then if we go back, I'll be able to see that protocol in my list when it might due, when it should start, and I'll be able to view it in that calendar as well. Another way that you can manage your work across across a lab is with the reagent manager. So again, if I go here, I can then go into manage reagents. So I think I already have that prepared as well. No, I don't. We'll go in there. And so I can see all of the reagents that are associated with my workspace. So when people are drafting protocols, they'll know what's available to them. So I can add new reagents in myself by searching for existing ones, and then they will be added to the list that's available within my workspace. Or I can create my own custom one by adding in this information here. If you do have an inventory list that you want to upload, you can do that too. So I'd have to go into this create a reagent window and then upload reagents. And then you can upload a CSV file in the following format. And then all of those reagents will be created and you can add them to your workspace. So we can go back. And then I can show you with these reagents as well that I can come in and I can edit the details of that workspace, the details of that reagent in the workspace. And so put in some information like where in the lab it's stored, when it was last opened, things like that, if you do want to manage it that way. And for a reagent as well, I'll be able to see what other published protocols also using that same reagent. So sodium chloride, pretty basic. So we can see, if I go back and I open reagent page, we can see the other protocols that are using that same reagent. So if you're having trouble with a reagent or a step that has that reagent, you might be able to see if other people are what other protocols are also using that and might be encountering the same issues, for example. And then lastly, to finish up, I will show you when you are publishing, you can publish a individual protocol. But if you have, for example, multiple protocols that might need to be followed in sequence, are associated with one another, or are all associated with a manuscript, then you might want to group them together. And you can do that by creating a new collection. So you can come up here and search new collection, or you can select the protocols that you want. So I think I've got two here. So we can see this is my collection. And you can shift select both of those and then go up and click on new collection. And so I can select where I want to save that. And then I'll be able to give that collection a name. And I can see the protocols within it. And so when this is all published, each individual protocol will have its own DOI, and then the protocol or the collection itself will also have a DOI. So people can cite, for example, the whole collection instead of citing a list of 5 different protocols within it. So that's all of the information that I had to go through on protocols.io. We've got a bit of time left. I will move back into the slides and finish up my presentation. Oh, one moment. All right. So I can obviously only show so much during a 1-hour webinar, but there's lots of places where you can get additional help. So you'll see some of them listed here on the slide. So you can refer back to that later when you receive these slides. And another thing just to think about is that it is a really nice time to trying to get more familiar with protocols today or learn more about it, because more and more journals are recommending to put your methods on places like Protocols Today when you are publishing with them. And these aren't just Springer Nature journals as well, but many others. And similar thing is happening with funders. So you can see here, these are funders who recommend in their policies that you post your methods and protocols on Protocols Today. But for example, this funder here, ASAP, they actually mandate that if they are funding your research, you are sharing those protocols publicly on Protocols Today. And if you are doing that, it makes sense to add in all of that additional functionality to those protocols as well. So that's all from me today. Thank you very much for listening. Before we go into the Q&A, I'd like to ask people to take a moment to fill out our webinar feedback survey. So it helps make sure that what we're talking about in these webinars is as relevant as possible for you. So I will put a link to that in the chat, or you can scan the QR code as well. And yeah, so the floor is open for questions. So if you do have any, please put them into the chat. Okay, I see something has come through. Okay, we've got Sergio. Would you mind sharing the view of your 96 well play Excel file? Yes, I'll do that right now. Um, one moment. So you should, I'm moving my windows around here, so let me pull it up. It is a pretty basic Excel file with some information into the cells. All right, here we go. Sorry, I'm normally on multiple screens, but I'm juggling, well, I'm only on one screen today. All right, there you go, Sergio. So you can see in here, if we think back to that plate map, you've got the negative controls, the positive controls, the information all stored in the cells. So it's relatively straightforward. It's pulled all of this text across and it's automatically detected the size of the well plate map and created that for you, but it hasn't pulled across the colors here. But if you did have, for example, additional information in your Excel file, for example, like headers, extra notes, you'd probably want to just copy out exactly what you want to bring into Protocols.io, and then save that in a separate file to upload. But I hope that helps you visualize how the information comes across. All right. Let me see. We've got another question. How can we integrate ethical standards disclosure within the metadata manager to ensure that indexing algorithms recognize institutional titration as both a technical and ethical framework for institutional reform? Um, I'm I'm not sure what you're trying to achieve here. If it's in terms of, I think if it's in terms of creating change across institutions on a broader level. Um, Obviously, there are many ways to do that that are outside of protocols.io. Could you provide maybe a little bit more information on what you're trying to achieve specifically, like within the layout of protocols.io and the files? Maybe I can help you answer that question a little bit better. You can, yeah, you can just type it into the chat as well. If anyone else has any other questions, what I can do too is I can stop the recording if anyone would prefer to ask their questions off of the recording. Otherwise, if you can share any more information, Abdurauf, I hope I'm pronouncing your name right, on what you're trying to achieve, I can try and help. All right. I don't see any other information coming through. If you are typing, just put typing into the chat and I'll wait. Otherwise, I might go ahead and finish up the session today. All right, then. Thank you everyone for joining. If you do have any additional questions, please don't hesitate to reach out to info@protocol.studio. I'll put that into the chat as well. And have a great day and have a great rest of your week. Bye bye now.